Okay, hello everybody. So this is our last lecture. It's the end of the microbiology course. Um, so we're going to wrap up with bacteriophage and then spend maybe 45 minutes, 40 minutes on viral infections in humans. Just a couple of examples of viral infections. I want to leave a little bit of time at the end so we can go through the final results of Microbe of the Week. So I will be aiming to finish talking at about, I don't know, 10:30 or maybe 10:35 in about 1 hour. Okay, so just to get everybody thinking a little bit, I would like you all to think about, kind of as an introduction to what we're going to do about bacteriophage. Last time on Monday, we looked a little bit at the lytic replication cycle in T4 phage. And before we go and do lambda and the lysogenic cycle, I just want everybody to think a little bit in more general terms about bacteriophage and their role in the biosphere. Okay, so first thing is to think about how many of them there are. Okay, so in seawater, in the aquatic environment, how many do you think there are in 1 ml? So just everybody put your hands up. Okay, who says like one phage, one PFU per ml? About 100. 10,000 per ml, 10 to the 4. We got about 8 people putting their hands up. 10 to the 6, 1 million. About the same number of people, about 100 million per ml, 3 or 4 people. Okay, well, yeah, it's around about in between these two, 10 to the 6 to 10 to the 8. If you're at the beach in coastal waters, this is a rich environment, then you might have 10 to the 8 per ml in coastal waters. In the deep ocean, probably it's going to be more like 10 to the 6 per ml, 3 times 10 to the 6 per ml. Now, the oceans are pretty big, right? There's a big volume there. So how many phage, okay, viruses or phage, both of them together we're talking about, but in fact, most of the virus particles that you will find in the ocean are bacteriophage, viruses that infect bacteria. So how many of these will there be altogether on the whole planet? All right. Okay, so you have 10 to the 10. That's too small, right? Obviously. 10 to the 20. Hands up if you think that's right. One person. 10 to the 30. 3, 4, 5 people. 10 to the 40. Whoa, most people think that's 10 to the 40. That's way too big, man. Man, that's just such a huge number. I don't even know if there are that many atoms on the planet Earth. All right. In fact, it's 10 to the 30. 10 to the 30 is the total number of virus particles in all the world's oceans. Or that's what the estimate is. But that's very, very difficult to visualize how big this number is. You know, if 10 to the 30, 10 to the 35, 10 to the 50, I can't visualize these numbers. So to try and visualize this, okay, you have to imagine that each one of these particles, okay, it's a virus particle about 100 nanometers in diameter, and each one of them has got maybe 0.2 femtograms of carbon. So of course, with 10 to the 30 and 0.2 femtograms, you could calculate the number of tons, but to make it more interesting, the question is, how many blue whales is that equivalent to? Okay, you can imagine a blue whale, it's big, it's massive, right? So if you put all these virus particles, all the carbon in them, how many blue whales would that be? Okay. Okay, one blue whale. Anybody say that? What, 100? Okay, a couple of people. 10,000 blue whales. Just try and imagine them all lined up. 1,000,000 blue whales. 4 or 5 people, and 100 million blue whales. Nobody thinks it's that much. In fact, it's almost 100 million. It's 75 million blue whales. That's the amount of biomass that is in the world's marine viruses. And just one other way of thinking about this is if you put them all end to end, they're really, really tiny, but if you put them all like one after the other, how long would it be? Okay, so 1 million kilometers, 1 light year, 1,000 light years, 10 million light years, or 1 billion light years. Okay, you just have to try and guess this one, all right? Although you could work it out if you're quick on your calculator. So 1 million kilometers long. 1 light year. That's pretty big, right? Yeah. 1,000 light years. 10 million light years. Anybody say that? One person. 1 billion light years. Oh, it's just too huge, right? Well, apparently the correct answer is 10 million light years. That's how long they would be. That's how long it would be, this line. So I always forget this from one year to the next, but apparently the Milky Way galaxy is only 10 to the 5 light years across. So it's 100 times bigger than the Milky Way galaxy. And the nearest galaxy to us is the Andromeda galaxy. That's only 2.5 million light years away. Okay, so that's the amount of marine viruses there are on Earth. Okay, it's a big number. So what are they doing, all these viruses? Well, it's kind of like the cold dark matter of the biosphere. Okay, normally when you think about carbon cycles, nitrogen cycles, you're thinking about primary producers and then some kind of organism that's going to graze on the primary producers and then some kind of predators. So you have a kind of ecological food web which is going to go from primary producers all the way up to predators. Some of these, in fact a lot in the ocean, are going to be cyanobacteria. Photosynthetic bacteria, right? They're going to be fixing carbon, fixing nitrogen, and producing organic carbon and nitrogen that can move up the trophic levels in the ocean. And what is going to happen is that bacteriophage are going to infect these cyanobacteria and lyse them. So this will release organic carbon and organic nitrogen. It's going to be free in the ocean. And this will be available to be used by what kind of trophic type of bacterium requires organic carbon? Heterotrophs, right? Bacteria again. But then these bacteria are also going to be infected and lysed by some bacteriophage. And that will also release organic carbon and nitrogen. So the net effect of all of these bacteriophages in the ocean, in other ecosystems, is that a lot of the organic carbon and nitrogen is not going to be available to move up through these trophic levels, but it's going to stay being shuttled around these lower levels in between primary producers, okay, autotrophs, litho-autotrophic bacteria, being lysed, releasing organic carbon. This is being used by heterotrophs. They're going to be lysed again. So a lot of the net effect of these huge amounts of phage in the ecosystem is to keep organic carbon and nitrogen down at the lower levels of the trophic web. And this is, a big thing because bacteriophage are the second biggest amount of biomass on the planet. So the biggest is going to be prokaryotes, bacteria, and the second biggest is viruses, bacteriophage. So they have a large effect on these, important biogeochemical cycles. The other thing that you'll need to bear in mind is, as we'll see for lambdaphage, sometimes bacteriophage can integrate their DNA into the DNA of the host. So all of these bacteriophage are a kind of huge database of genetic variability that's being carried outside of cells and being exchanged between bacteria. So there's a lot of genetic variability in bacteria, but there's much more in bacteriophage, and sometimes it switches between the two. So that's another important role of bacteriophage in the biosphere. Okay, so there's a lot of them quantitatively, and when you look at one species of bacterium, like Escherichia coli, you can find several different types of phage which can infect the same bacterium. So you can easily have, I don't know, so T2, T4, T6, very quite similar, but these T-odd phage are different families. Lambda M13, all these ones, T2 and T1, T3, T7, and lambda, they are all cordovirales, double-stranded DNA genomes, single-stranded DNA genomes here, RNA genomes here. So you can easily have like 10 different species of phage infecting one species of bacterium. So there's more biodiversity in the virus world than there is in the bacterial world. And when you look at different types of bacteria, wherever you look, you can always find some kind of bacteriophage that infects all those bacteria. And that's true for, quite ordinary bacteria, gram-positives, gram-negatives, also true for mycobacteria, and it's also true for archaea. So whatever kind of cell exists, you have some viruses that can infect and parasitize that cell type. Okay, so there's a huge amount, quite a lot of diversity, but rather fortunately for us perhaps is that the most frequent or the dominant types of bacteriophage are these cordovirales, that is double-stranded DNA bacteriophage with an icosahedral head and some kind of helical tail structure. So the caudal, okay, that's for tail. And there are, so this is an order, cordovirales, and there are 3 families of viruses, the Myoviridae, Cephaloviridea, and Podoviridae. So Myoviridae, this is T4 bacteriophage. This is the classic one. It's characterized by a long contractile tail, so this is what will inject the DNA into the host cell. Cephaloviridea, like bacteriophage lambda, they have a long, flexible, non-contractile tail. And then the Podoviridae, like phage T7, that's what you have here. You have an icosahedral head and then a kind of like base plate at one of the vertices of the icosahedron. So it's like they don't have a whole tail here. They've just got like this basal structure attached directly to the head. Okay, so Myoviridae, like T4, are mostly lytic. Okay. they seem to be able to infect more than one bacterial host. Podoviridae, these guys here, also they are lytic, but they seem to be very specific for one bacterial host. Whereas Lambda and the other Siphoviridae, they can also have a lysogenic cycle. So before we see what that means, okay, just Cordoviralis, this is a dominant type of bacteriophage. If you take, 10 ml of water from the beach at Pornic and you have a look at the virus particles that are floating around there, most of them will be Cordoviralis. However, if you look in different environments or have a look at the rarer forms, you can see that it's not the only type of bacteriophage that exists. You can find filamentous phage, you can find icosahedric bacteria particles without any tails. And if you look in rather unusual environments like hot geothermal springs at a low pH, then you find these weird forms here. Guttavirus, Lipothrix, it looks like a kind of snake here. Look at the size of this thing. This is 100 nanometers here, so it's easily, 700 nanometers long, maybe a micron. Fusella viridae. So there are unusual types of bacteriophage, and we know a lot of things about the biology of the Cordoviralis because they're very frequent. They've been studied intensively for, maybe more than 50 years now. And these ones, we know a lot less about them because they've only been discovered in about the last 10 or 15 years. Okay, so let's just go back to phage lambda, the Siphoviridae, and lysogeny. What exactly is that? Now, I'm going to show you a short video here. And this is, so these are bacterial cells, these are E. coli, and they're being infected by phage lambda that's been genetically modified so that infected cells will express the green fluorescent protein, GFP. Okay, so this is light microscopy. You can't see the viruses, they're too small. You can only see the cells, and when they get infected, they're going to become green. Okay, so let's have a look here and hope that it can. Oh. Yeah, well, something might happen, it might not happen. Okay, do you see that? So you had one cell up there, it got infected, it went green, and then it was lysed. Okay, so this is a lytic cycle. And what's happened to this one? This one's also infected. It's gone green, right? Okay, but in fact it carries on living and it carries on growing and dividing. Okay, so you had two very different results of infection in these bacteria. The one that was the one that was up here, okay, it was infected, it was lysed, it was killed. And this one down here, it's infected, but just carries on living. And this is what's called a lysogenic cycle. So what is happening in these two cases? Okay, so for a lytic cycle, bacteriophage lambda will kind of start out just like a bacteriophage T4. So it attaches to the surface of the cell, the DNA is going to be injected, it will circularize, start to express the bacteriophage proteins, replicate its DNA, produce viral particles, and then lyse the cell. However, in the lysogenic cycle, what's happening in this cell here Okay, so that's this cell. It's going to have a lytic cycle. This one is going to have a lysogenic cycle. So it starts off the same way, okay? Attachment, entry, decapsidation. So the phage DNA enters the host cell. It will circularize. And then this time, instead of replicating the phage DNA, it's going to insert itself into the bacterial chromosome. And once it's been inserted there, then some of the phage genes can still be expressed. In this case, the green fluorescent protein, which is why these cells are green. Now, in this case, late genes of the phage are not going to be expressed. So you don't get the production of virus particles and you don't get lysis. That's why these cells survive. So this cell will carry on growing and dividing. Every time it divides, it replicates its own genome, the bacterial chromosome, but it's also replicating a copy of the viral genome. So this virus genome, or the phage genome, incorporated into the bacterial chromosome is called a prophage. Yes? No, it's regulated, and we're coming up to that in the next slide. But that's the important question. Yeah. Okay, so another thing before we go on to look at this, another thing to remember is this is reversible. Okay, so once a prophage is integrated into the chromosome, later on, if one of these daughter cells is stressed by heat shock or maybe antibiotic treatment or UV irradiation, then it can excise from the chromosome and reactivate a lytic cycle. Excise from the chromosome, produce more phage, and lyse the cell. So these, what so-called temperate phages, kind of alternate between lytic and lysogenic cycles. So there are two different strategies for, well, ultimately just replicating the phage genome. Okay, so how is this controlled? Is it just random, or is it logical in some kind of way? Well, it's a decision or molecular kind of decision that's made very early after infection. Okay, so the earliest genes that are transcribed after phage lambda infection are N and CRO, and then Q and C2. And it's these two that play an important role in deciding whether the phage is going to become lytic or lysogenic. So in most cases, C2 is going to be degraded. So you only have CRO, all right? CRO and N and Q. And this will allow progression through the lytic cycle. But in some cases, C2 will survive, I guess, or persist. And then what it's going to do is it only allows transcription of the integrase gene. So the phage genome is integrated into the bacterial chromosome. And then it allows expression of C1. So C1 is going to shut down the expression of the late genes. So the phage doesn't lyse the host cell. So this is the key event, okay? This C2 protein, whether it's degraded or not by the host cell proteases. And the key to this is understanding what conditions allow C2 to persist. Generally, it's conditions that are not favorable for bacterial growth. So low temperature, or if the bacteria are in the stationary phase of the growth curve. In those cases, there'll be lower metabolic activity, so the host proteases will have lower activity. This favors C2 resistance to degradation and favors lysogeny. The other condition is if you have several bacteriophage infecting the same cell at the same time. So each of them will start to express C2, and there's going to be too much for the host cell to degrade all at the same time. So this last point is really quite clever from the phage point of view. Okay? Think about it. Okay, if you have a situation where you have lots of host cells, one phage comes on here. If all of these bacteria are growing very nicely, they're in good conditions for growth, one phage infects a cell. Then it will undergo a lytic cycle, right? So it will release a lot of phage and it will go and infect all these other cells. However, if you've got a situation where you only have very few bacteria and a lot of phage, all of them are going to infect the same cell at the same time. Now in these conditions, if you start a lytic cycle, it's a very bad idea, because once these bacteria have been lysed, you release other phage and there's no other host to infect, because there's already far too many viruses around for the number of host cells. So it's not a good idea to have a lytic cycle. So in this case, it's a smart move to integrate into the host cell chromosome, and in fact this renders the bacteria resistant to lysis from the other phage. They become resistant to infection. And then they can survive and grow, and then, later on the phage can excise and go back to a lytic cycle. So this, every time you find something in biology that is complicated, well, it's not just an accident, there's a reason for it. Okay, so the reason here is that it gives the phage another strategy to replicate itself. Okay, so that's looking at things from the virus point of view. From the human point of view, this is a problem because many of these temperate lysogenic phages carry bacterial virulence factors on their genomes. So they can integrate into bacterial chromosomes, and they all carry virulence factors with them. So a lot of different exotoxins are coded on bacterial prophage. So for example, in enterohemorrhagic E. coli, the Shiga toxins are coded on two integrated prophage, SP5 and SP15. And you can see that on this E. coli genome, there's quite a lot of lambda-like prophages that have been integrated into the chromosome here. Now, the net result of this is that bacterium to another through lysogenic or temperate phages can convert commensal bacteria into pathogens. So just a harmless commensal Escherichia coli could be infected by a bacteriophage that carries genes for virulence factors. And if they accumulate them, and if they accumulate virulence plasmids, then they can become more and more pathogenic. Now, this is possible between bacteria of the same species, and in fact, it's also possible for phage to transfer genes between bacterial species. So in this paper, they showed that it was possible to transfer genes from Staphylococcus aureus to Listeria monocytogenes, two quite different gram-positive bacteria. Okay, so that's about it for bacteriophage. Before wrapping up, let's see if you remember something from Monday about phage T4. And the lytic cycle. Okay, so function of the late genes are 1. Control the expression of phage genes. Anybody say that? 2. Control phage DNA replication. Structural proteins, who says that? Yeah, one person. You're right. Lysis of the host cell. Yeah, okay. So late genes, early genes, okay. Okay, so for replication, okay, remember these steps: attachment, entry, decapsidation, and then choice between a lytic and a lysogenic cycle. Which phage has a lytic infection, which phage has a lysogenic cycle? And try and remember or try and understand why lambda phage has such a complicated system. Okay, what is it gaining from doing this? And the overall summary for phages, there are tons of them. Just because there's so many, they must be doing something important. But mostly, their importance for microbial ecology is not really very well understood. Something I didn't go into, but historically, phage have been very, very important for understanding really the fundamental processes of molecular biology: DNA replication, control of gene expression, the nature of the genetic code. This kind of stuff was all found out by working with bacteriophage. Other things extremely important for molecular biology: restriction enzymes and the CRISPR-Cas9 system, which is used in gene editing technology now, found by looking at bacteriophage. And the last thing, which has disappeared, is that, okay, remember their importance in human biology, as they are the vectors that carry virulence genes and shuttle them between bacteria. Okay, so we have about half an hour now to try and look at viral infections in humans. So firstly, okay, when people say, "Oh, I had a virus last week. I was a bit ill," what you're thinking about is this kind of acute self-limiting infection. This is what you get most of the time. So this could be rhinovirus infection, adenovirus, or gastrointestinal viral infection. So what happens is you'll have infection. A few days later, you'll have a peak of viral replication, and then you'll have symptoms, and then you get better. Okay, acute self-limiting infection. As well as that, you have some acute, very severe infections, where you'll have a infection, peak of viral replication, symptoms that get worse and worse, and then finally you can die. So the examples of this would be something like rabies or Ebola virus, where a large number of people a high percentage of the people who get infected will actually be killed. So among these acute infections, you've got some that are, pretty much benign, and those that are very, very severe. And then you have others that are in between. Now, other viral infections can be persistent, and among persistent infections, there are latent and chronic infections. So chronic infections, you have infection, a peak of viral replication, then symptoms that seem to, people recover from the primary infection. But the virus is never really cleared. It never really disappears. There's always ongoing viral replication. And so virus replication continues. Symptoms come back from time to time, and gradually they kind of get worse. Okay, but this whole process can take decades. So like the hepatitis viruses, hepatitis B, hepatitis C, HIV infection, these are examples of chronic viral infections. And if these aren't treated, then they have a tendency to get worse and worse and worse and more severe, and people will end up dying. Of course, it takes a long time. So for hepatitis C, for example, maybe half the people who have untreated hepatitis C, in chronic hepatitis C, half of them will die of their hepatitis C. The other half will be killed by, cancer, heart attacks, car accidents, this kind of stuff, because these will happen before the chronic infection has had enough time to actually kill you. And then next to that, you have latent infection. So here you've got the primary infection, viral replication, symptoms. Then the virus seems to disappear completely but it persists latently in some kind of site in the body. And later on, decades later, you can have reactivations. So virus replication is reactivated, you get symptoms again, and then it goes away, then it comes back. So these are herpes viruses, are the big classics for that. Herpes simplex, varicella virus, okay. So if you have a zona when you are 85 years old, it's probably the same virus that infected you when you were 2 years old. And it's persisted latently all through your life. Okay, so acute, persistent, two different types of infections. Okay, how do you catch viruses? Bit like bacteria, really. Mostly it's mucosal surfaces are the most susceptible sites for viral infection. So gastrointestinal, respiratory, genital. Skin, like for bacteria, it's quite rare. Skin is quite a good barrier, quite an effective barrier that prevents viral infections. Of course, you can have viruses that cross the skin through insect bites or tick bites. And blood, of course, can be a direct mechanism of transfer of viruses. And last but not least, okay, mother-to-child transmission can be very, very important for some viruses. So, for example, hepatitis B virus. Everybody knows it's blood-borne, but this is not a very natural route of infection for a virus. It depends on medical technology. So, for the vast time of this virus's existence in the human population, yeah. Yes, I think that's the reason, and it's also the suspicion that there may be viruses we haven't discovered yet, or early in your life, maybe the tests weren't done on all the viruses we know about now. Okay, so for hepatitis B, the real motor that maintains a large number of people with chronic hepatitis B, especially in Southern Africa and Asia, is mother-to-child transmission. So this can occur during pregnancy in utero, but almost more frequently during birth, because you have contact of the baby's mucosa with mother's blood, for example. Okay, so you can have access from the infectious biological fluids to the baby's mucosa, in the mouth, even around respiratory as well. And breastfeeding can also be a vector for some viruses. Okay, so the virus has arrived. Afterwards, it infects you at the site. There's two possibilities. Infections can either be local or systemic. So in a local infection, replication is going to be restricted to the initial site of infection. You won't find viremia, that is virus in the blood. And mostly these infections have a short incubation period. The incubation period is the time between infection, day 0, and when you have symptoms appear. Okay, so it can be 24, 48, 72 hours, that sort of thing. So examples of this: rhinovirus, you catch a cold. Rotavirus, you get viral diarrhea. Okay, site of infection, gut mucosa, symptoms in the gut. Systemic infection is a bit different. Okay, in this case, the target organ, that's where you're going to see symptoms, is distinct from the site of infection. In order to travel from the site of infection to the target organ, the virus has got to pass through the blood, so you have viremia. And this takes a bit longer, okay, so the incubation time can be intermediate or long. So one example, for example, hepatitis A virus, site of infection in the gut, symptoms in the liver. Measles, the initial site of infection will be the respiratory mucosa, and the symptoms are on the skin. Okay. HIV, infection in the genital mucosa. Yeah. Well, kind of. That's the definition of the incubation period. The incubation period is when you have no symptoms. All right. Then you start to have symptoms, but they can be more or less severe and more or less specific. So the first symptoms can be very non-specific. You have a fever or something like that, and then you might have other symptoms that are more pathognomic. Okay. Okay, so in the time we have left, yeah, about 15 or 20 minutes, I want to look at two specific infections. Two acute infections, one which is localized and one which is systemic. So our example for localized acute infection will be influenza virus. And it looks like this. So it's negative strand RNA virus. The genome has 8 segments, like this. So each molecule of RNA pretty much codes for one gene in the viral genome. You can think of this as being like a virus that has got 8 chromosomes. Okay, it's got a segmented genome. Okay, these genome segments are arranged with a kind of helical nucleocapsid, so in a kind of a stick shape, and they are all packaged together in an enveloped virus particle. So here's the envelope, and there are 2 viral glycoproteins in the envelope: neuraminidase, which is important for viral release, and the hemagglutinin, which is essential for attachment. So hemagglutinin is a viral attachment glycoprotein. So if you catch influenza, this is what happens. So day 0, you get infected in the respiratory mucosa, mostly the upper respiratory tract. Very quickly, you'll have symptoms: fever, tiredness, muscle pain, headache, maybe a cough, not always. Symptoms persist for 3 to 5 days, and then you can feel quite bad for maybe the next couple of weeks. And that's in a kind of normal, self-limiting, non-complicated form. However, things can get worse. If you start off with influenza, then it can be complicated by a bacterial infection, or if the virus leaves the upper respiratory tract, comes down into the lungs, you can get pneumonia, and this can be more serious. Okay, so people who are, so overall, this for seasonal influenza, the case fatality ratio, that is the number of people who get killed after an infection, is less than 0.1%. So less than 1 person in 1,000 will be killed by a seasonal influenza infection. And those people will be the ones who are most susceptible. Older people, young children with a kind of predisposition for severe infections like asthma, other lung conditions. So the susceptible population, older people, children with asthma, have to get vaccinated every year. So my son, he gets an influenza jab every year because he needs it. Also, 30% of infections will be asymptomatic in healthy young adults. Okay, so this is very important for prevention. That means that hospital staff, people who are in contact with the susceptible population, should get vaccinated, because they might be infectious even if they don't know they're infected, because they have no symptoms, and they can still pass on the virus. Okay, so this is the kind of segment of the population that is at risk here: older people and people with pre-existing conditions. Now, vaccination, there's a problem with influenza vaccination because the virus is variable. It's an RNA virus. It's got a high mutation rate. And over time, the hemagglutinin protein changes because it's a kind of evolutionary arms race between the host population and the virus. So the virus mutates to escape the immune response of the host, and over time, it doesn't stay fixed. So that means that the vaccine has got to be changed regularly. Okay, so this figure only goes up to 2002, but you can see that every couple of years, the vaccine's got to be changed. And the efficacy of the vaccine will be variable from year to year, depending on what viruses are circulating. So you get a lot of people saying, oh, you know, I got vaccinated, but I still got influenza. Well, you're kind of unlucky because that means the virus that infected you was different from the vaccine strain. Okay, so that's seasonal influenza. It's a real health problem, but manageable. And the thing that is worrying about influenza are the pandemics. So every few decades, there's a new influenza virus that will appear in the human population, apparently from nowhere, and will start circulating. So because it's an antigenically new virus, the whole world population can be susceptible, and you'll have a very large number of infections that particular year. So there have been several of these events. The last one was in 2009, kind of Mexican influenza. And this one was not very dangerous. It was less pathogenic than the seasonal flu. And at the other end of the spectrum, you've got something like the Spanish influenza here. Probably killed, I don't know, 25 to 50 million people over a 2-year period. More people than were killed in the First World War. And this virus had maybe a 5% case fatality rate. So a lot of otherwise healthy young adults were getting killed by this virus here. So where do these pandemic viruses come from? Okay, either, okay, two sources of these new viruses. Either it's the transfer of an avian virus to humans, so the bird virus becomes adapted to a new host, or it's a kind of genetic mixing between avian and mammalian viruses. So how does this occur? Well, it's because of this segmented genome. Okay, so influenza virus has got, each one has got 8 genomic segments, and if you've got a human virus and a bird virus that infect the same cell in the same person, both of these viruses will start replicating. So you'll have, you know, avian virus genome segments and human virus genome segments in the same cell. And from time to time, you'll have viruses that have packaged some avian gene segments and some human gene segments. So then you can get a mosaic virus. This is not really recombination, it's reassortment. And this virus may be able to replicate very efficiently in human cells, but it will be entirely new and may be very pathogenic. So this is a recurrent problem for influenza virus surveillance. That's why every time there's a new highly pathogenic avian flu, there's a lot of surveillance, because this kind of event is going to be is very rare, but it's happened before, and every time it happens, you can't really predict what the results are going to be, whether the virus is going to be very pathogenic or pretty harmless. So everyone's waiting for, you know, when is the next influenza pandemic going to come? So that's our first example, and second example is Ebola virus. Okay, it's another negative strand RNA virus. This time the genome is in 1 molecule, not 8 segments. Once again, it's got a helical nucleocapsid, and it's an envelope virus. Okay, so because of the helical form of the nucleocapsid and the large genome size, it produces these filamentous particles. Looks like this. Okay. If you look at the genome, what have we got here? Okay, L. This is the RNA polymerase, so the virus use it for replicating its genome. NP, the nuclear protein, so that makes the nucleocapsid. And GP, that's the surface glycoprotein. It's on the outside. So GP and NP, these are the structural proteins. L, that's for virus replication. And then you have these other accessory proteins, VP35, 40, and so on. Now, I don't claim that I know the functions of all these proteins, but VP35 and VP24 are very important for pathology because they will inhibit the host interferon response. Another thing that you need to know here is, okay, the glycoprotein, if you have antibodies against the glycoprotein, that will block infection. So people who have survived, well, we don't really know for sure, but probably are immune. And all the vaccines that are under development against Ebola virus are based on inducing antibodies against the surface glycoprotein. And these are recombinant viruses, other types of harmless viruses, and you stick the glycoprotein of the Ebola virus on the surface, the virus can replicate a little bit, doesn't cause illness, and you'll be immune against Ebola virus. So these have all been successful in primates, non-human primates, and probably this one is the most promising candidate for development. Now, one of the reasons why this is probably going to work and will be effective over a long time period is because, in fact, Ebola viruses are quite stable through time genetically. And that's because they don't circulate continually in the human population. So influenza virus, we just saw, it changes genetically over time. This virus is always circulating in the human population. So there's a kind of like, as I said, it's an evolutionary competition between the host and the virus. And the virus survives by mutating. But Ebola virus doesn't do that because it's not a real human pathogen. Okay. Yeah, I'm just going to skip this one. So if it's not circulating in humans, where does this virus come from? Now, it seems like the natural host of these filoviruses are bats. And the virus is going to be adapting to its natural host, the bat. And from time to time, these bats can transfer infection to other animals, or even, people can hunt these bats and catch them and eat them and become exposed to infectious material in the bats and become infected. Maybe actually eating these animals is not necessary. I saw a great talk at the European Congress of Virology last month by people who looked at Marburg virus infection. It's another one of these highly pathogenic filoviruses like Ebola. And what had happened is that there was a mine, in some caves in Central Africa. And these caves were the home for about 100,000 bats in a colony. They were infected by Marburg virus. And the virus was excreted in their, in their feces, in their urine. And when the bats are flying about, they're ejecting all of this stuff. So from time to time, miners and tourists who went to visit the caves to see the bats could just be exposed to the virus because of the bats are flying by and they're bombarding them with infectious material. So you don't even need to, catch one of these things and eat it. You just have to be nearby. And just after, the virus has been released, you get some of it on your hair or your hands, and then you bring it to your mouth, and then you infect yourself. So sometimes you get these sporadic transmission of the virus from the natural host population, which is the bat, and it can infect people. And then once it's in the human population, then you can have human-to-human transmission. So this type of pattern is called a zoonosis. So it's an animal virus being transmitted to humans. Okay, so what will happen to you if you catch Ebola virus, Zaire Ebola virus? Okay, so there are different species of Ebola virus depending on the country or the region where they were originally identified. So they're more or less pathogenic. Zaire Ebola virus is the worst one. Okay, so in human-to-human transmission, okay, infections via mucosa, maybe the mouth, or even just by cuts and abrasions on your fingers if you're coming into contact with infectious material. The eye as well can be a site of infection. So this is why if you saw pictures of nurses, doctors working with in the Ebola virus epidemic in West Africa, they had this like full body covering, you know, with a full face mask to prevent transmission through the eye mucosa. Then you'll have a maybe 2-day to 3-week incubation phase. The average is 8 to 10 days. Okay, let's say 1 to 2 weeks. Then you'll have early symptoms, very, very non-specific. Okay, you'll feel tired, headache, fever, vomiting, diarrhea. This could be a lot of different things. So very early in an epidemic, this can be misdiagnosed. So for example, depending on the country, you know, this one might be misdiagnosed as malaria or typhoid fever, or or you know, bad case of influenza, something like that. So this will last for 2 to 3 days, and then things get progressively worse. So you'll have people with you know, the fever doesn't go down; it gets more and more severe. People will become, you know, completely passive, you know, just lying down. They don't move. They don't answer questions. And then you have the hemorrhagic syndrome, so bloody vomit, bleeding from the nose, mouth, eyes, anus. And then people will have internal hemorrhages and die. And it happens very quickly. Okay, so from here to being dead, the average is about 8 days. And the case fatality ratio for Zaire Ebola virus is something between 50 to 70%. So it's very, very highly pathogenic virus. So how does it do this? Why is it such an effective killer? So what is happening is, okay, you get the initial site of infection somewhere at the mucosa or at the skin, transcutaneous infection. So the virus is going to start infecting cells in the body and will disseminate throughout the body, replicate to high levels in the liver, in the spleen, in the lungs. And all of this time, throughout the 1-week incubation phase, it's like your immune system has gone to sleep. Okay, you don't recognize you've been infected. So this is the VP24, VP35 proteins. They're inhibiting the host interferon response. You're walking around, you know, thinking perfectly fine, and in fact, your whole body has been, well, is turned into a virus replication factory. Then later on, you've got a very high viral load, and then finally, your immune system wakes up, okay? And then your monocytes and macrophages get hugely activated. It's just, oh my God, we've got a virus here, we've got a virus everywhere. And you get a huge stimulation of, these monocytes, macrophages. They'll start releasing TNF-alpha, interleukins, tissue factors. So this is going to give you the high fever, cause vascular leakage at the endothelial cells, cause intravascular coagulation, and that's what gives you the hemorrhagic syndrome, and that's what's going to kill you. Now, does this ring any bells, all of this? Maybe I didn't talk about this enough. So in terms of pathology, it's really quite similar to toxic shock. Okay, so if you've got endotoxin, bacterial endotoxin, with a large scale systemic infection of gram-negative bacteria, you'll have a similar kind of thing. You'll have lots of your monocytes, macrophages getting activated all over your body. Okay, either through endotoxin in the case of a bacterial infection or because of Ebola virus here. And this gives you, you know, quite a similar kind of syndrome. Okay, so Ebola, so that's the pathology. Now, how does the virus transmit from one human to another? It's not a normal human infection. Well, direct mode of transmission. There's no indirect transmission. It's not transmitted by mosquitoes or you know, contaminated water or something. There needs to be direct contact with infected body fluids. Could be blood, tears, saliva, breast milk, sweat. Okay. Now, the level of contagious risk is proportional to the viral load. Okay, so the more virus that is in the body fluids of the infected person, the more contagious that person is. So during the asymptomatic phase, viral load is quite low, so the risk is low. And then as people become more and more symptomatic, it's because they have more and more virus, so they become more and more infectious. One of the things that was very, very problematic in the West Africa outbreak, this was 2014, 2015, is that the dead bodies remained infected. So in the West Africa outbreak, there was about 30,000 cases overall, and you know, maybe 15,000 to 20,000 people were killed. And the only reason we still don't have a raging epidemic is because there was a big international effort to control these transmission chains. And this was by far the biggest Ebola virus epidemic ever, because all the other ones, the next biggest one had something like 300 or 400 cases. So it's like 100 times more severe than any of the previous known Ebola outbreaks. So as I said, going back to this, corpses remain infectious. And this was a big problem because when people died of this infection, they had a funeral, and the tradition were the body would be open and people would come up and you know, hug the person, kiss the person for the last time before they were buried. And then many people got infected at funerals. So one of the key elements to controlling the Ebola outbreak was identifying the contacts of all the people who were infected, trying to put them under observation as soon as they became symptomatic, put them into isolation, and to try and teach people about avoiding these modes of transmission. One of the very important things was putting in place respectful and safe burials. Okay, last thing about transmission here. Yeah, we've got. Now I'm going to skip this one. It's not really important. The only things I said, okay, it's not really airborne. Okay, so if you're standing next to someone who's infected and they're bleeding and coughing, of course they can project a kind of aerosol of infectious material, and you can be infected, you know, a couple of meters away, but it's not really airborne in the sense that it can't, you know, go through the air, and if I was infected here, I couldn't infect someone else at the other side of the room. It's just, you know, kind of limited droplet transmission. Okay, so for viral infections, just to round up here, don't forget you have diversity of viral infections, acute, persistent, local, systemic. The clinical outcome is not the same, even for the same virus. Okay, influenza virus infection, you go from 30% of infections will be asymptomatic. Maybe 1 in 10,000 will be a fatal infection. Depends on the variability of the virus, depends also on the type of person who's infected, host factors. Okay, and these are the main points of the 2 different infections we looked at. Okay, so that's it for the lecture. Now I'm going to keep, so we have 12 minutes before the end of the lecture here, and it's 2 things that need to be done. First one is final results for microbe of the week.